Kia ora and welcome. Our topic today covers two areas many of you have expressed interest in hearing more about: climate change and soil microbiology. What a mixture. Manpreet Dami is a senior researcher in molecular ecology at Manaki Whenua Landcare Research. Her research focuses on the interface of ecology and evolution using microbial systems and communities to simulate evolution in real time. This knowledge is applied to better understanding of pollination, the development of microbial biofuels, and the detection of unwanted pests. In this seminar, Manpreet will outline research which harnesses New Zealand's natural geothermal environment to create a laboratory in a landscape to evaluate the performance of soil microbes over a temperature gradient. I know you will have questions for Manpreet, and we will come to those at the end, but do put your questions in the question box as soon as you can so that we can manage that part of the webinar. That's enough from me. Over to you, Manpreet.
Thank you, Christine. Kia ora koutou. Ko Manpreet Dami tōku ingoa, ki Manaki Whenua ahau e mahi ana. Good morning, everyone. My name is Manpreet Dami, and I'm a senior scientist at Manaki Whenua Landcare Research. Thank you for coming along. Today, I will talk about why we need to know more about microbial respiration and how our natural geothermal hotspots provide the perfect experiments and what it is that they tell us. While it's only me here today talking about this work, it's a team effort that couldn't be accomplished without a dedicated group of researchers from New Zealand and abroad, as well as the support from Tauhara Middle 15 Trust, Tu Wharetoa Māori Trust, and OPP Farm. Respiration from soil is a major contributor of atmospheric carbon dioxide, thereby regulating the global carbon cycle. It is constituted by respiration from soil organisms, plant roots, and microbes. Just about all models of carbon cycling predict that in the future, more carbon will be lost to respiration, and that carbon stocks will decline globally. While we don't know how accurate our predictions are, just about none of the models incorporate microbial respiration. Microbes respire an estimated 60 petagrams of carbon dioxide annually. Microbes take up leaf litter and root inputs in the form of simple and complex compounds and move that through microbial factories to produce stable forms of carbon that is stored. At the same time, they lose carbon dioxide in the form of respiration, and this is the carbon dioxide that is lost to the atmosphere. As microbes respire, they supply numerous functions: nutrient cycling, increased water holding capacity, plant growth, primary productivity, carbon sequestration are just a few of them. So, if respiration is so important, what controls it? Microbial respiration is affected by many things, including moisture, type of carbon, and importantly, temperature. Temperature, as we know, has been on the rise for a while now and is predicted to continue rising for another good while. So what happens to microbial respiration under warming? Well, we know from a variety of incubation studies that when you heat up the soil, microbial respiration increases. And as you keep heating the soil, the capacity of microbes to keep respiring goes down. This feature of microbial respiration is called thermal sensitivity, or Q10, which is a parameter that tells us about how microbes might respond to warming. To reiterate, as microbes get warmer, their ability to continue respiring under further warming goes down. This is their thermal sensitivity. Or this is what we know, at least in the short term. But in the long term, we have guessed that this continued heating will overall reduce the sensitivity of microbes to temperature. So they will be less able to continue respiring at higher temperatures, such that in a warmer future environment. This hypothesis was put together by Bradford and colleagues in 2008. This idea specifically is called the compensatory thermal adaptation hypothesis. And a key expectation here is that the thermal sensitivity, the Q10 parameter, would decrease in the future, and that the microbial communities themselves might also change, they might evolve. And finally, that their growth will be optimum at a higher temperature, such that their T optima will also increase in the future. And part of this is due to evolutionary adaptation. We expect that the enzymes would change so that they're less affected by temperature, and we guess that's what the future of microbial respiration would look like. This predicted scenario may not be so bad from a total carbon dioxide loss point of view, because less respiration might mean less carbon dioxide. But what about the reality? Well, predicting the future is difficult because recreating the future is hard. Researchers have tried to simulate future temperature regimes using latitudinal gradients, for example, comparing the North and the South Island location. Temperature is different, but so is rainfall, pH, soil type, vegetation, and any number of factors. Similarly, with an elevation gradient, comparing the top of the mountain to the bottom. Again, vegetation is a lot sparser at the top. Soil layer is a lot thicker towards the bottom. This makes it very difficult to disentangle the effects of warming from everything else. And in experimental studies where you incubate the soil in a laboratory condition, there's a lot of variation, is what is often meant by long term, from months to years to decades. As a result, there is no consensus on whether microbial respiration will go down as per the hypothesis, stay the same, or even go up. In fact, some of the interpretation of the results from the original study that hypothesized the compensatory respiration has also been fiercely debated. As this debate continues to heat up, we turn to geothermal hotspots in our own backyard. Anyone who's done a road trip through Central North Island has probably detoured through the many of the world-famous geothermal wonders along the Thermal Explorer Highway. In addition to being excellent photo ops, as well as some of the most sacred hapu sites and cultural centers in the country, these hotspots are also natural long-term warming experiments. Within a few meters at the Wairakei Tauhara steam-heated depression, which is approximately 150 years old, we observed a wide temperature range, over 30 degrees Celsius, while other factors such as soil type, vegetation, rainfall, pH see much smaller variation. With this geothermal hotspot at the center, we measured the soil microbial community and its respiration. We collected samples at 3 different distances from the steam-heated depression, with the closest distance being warmer to simulate a future community, and further away being cooler to simulate more similar to a present community. We collected soils at 2 different depths. That gives us an idea of 2 different communities within each soil core. And within the same site, we were able to replicate the experiment under 2 types of vegetation. As you can see in this photo over here, on this side of the steam-heated depression, we have grassland-covered soil. And on the further away side from the steam-heated depression, we have soils that covered with prostate kanuga. At these sites, we looked for 3 features that represent the thermal adaptation hypothesis. The Q10, which is the temperature sensitivity of respiration, the microbial community composition, and the optimum growth temperature. And I will discuss the findings of each of these features one by one. For Q10, where we looked for the ability of microbes to compensate for warming and reduce their respiration, we saw no evidence. Basically, the thermal sensitivity of communities from the cooler sites in blue and the warmer sites in yellow was not so different. But despite that, we saw that the microbes themselves had changed a lot. The communities in the warmer and cooler sites, so cooler sites here, warmer sites here, were actually quite different. And we saw this both for bacteria and for fungi. For example, in this bottom plot here, what you see is the composition of the fungal communities in one of the sites. And as the temperature goes from cooler to warmer, we see a shift. We see that this pink group of fungi, which is a thermotolerant fungi, starts to completely dominate the community. We saw this trend under both vegetation types. Further, we did not see a shift in the optimal growth temperature, or T optimum, either under the warming regime. However, so for example, here we have T optima under various temperatures in the site, and you see almost no relationship. However, we did see that respiration itself was somewhat constrained by available organic carbon and moisture. So organic carbon is in percentage values here, and you can see that respiration overall goes up as carbon availability increases, while this effect is not controlled by temperature, which is in the different colors. There's a slight relationship with water availability. Water availability here is in the form of the size of the points. And you can see the smaller size points are clustered towards the low end of respiration, and the larger size points are spread out on the higher end of respiration. So what does this mean for our future warmed microbial communities? These trends indicate that overall the future communities might behave somewhat like current communities, even though they may look very different. To summarize, we do not see the compensatory reduction in the sensitivity of microbial respiration as was hypothesized from the communities that have experienced long-term warming at the geothermal hotspots. Based on this result, it could mean that microbial respiration might continue unabated in the future soil communities. But a shift in the microbial community composition suggests that these microbes will continue to evolve in the face of global warming. This study, with all of its gory details, is now published in Global Change Biology, and we will circulate a link to the publication after the seminar. Now, what could this mean for all of the functions that microbes play? These are some of the questions we are planning to address next. How does microbial biomass contribute to water holding capacities at these sites? We know that these microbes can support grasses and some specific plants, such as prostate canica. What does that mean for climate impacts for some species that may be buffered? That some plant microbe relationships might persist. The earlier example of the pink group of fungi that I had highlighted is a species called Physolithus, which is a fungal symbiont of prostate canica. So it is possible that plant microbe relationships may allow some plants to persist, or vice versa, some plants might encourage certain microbes to persist. And what about the carbon stocks. We all care about our carbon stocks. New Zealand boasts an average of 90 tons per hectare of soil carbon stocks nationwide. Will the potential of carbon sequestration in our soils change as a result of a shift in microbial communities? Will total soil respiration predictions increase when we include microbial respiration? How would that change our predictions of carbon losses globally? As we continue to add more and more carbon dioxide to the atmosphere and unbalancing the global carbon cycles, we have got to get better at understanding how microbes will change in the future so that we can better predict how they might change the future. And I will leave you with that thought. Thank you everyone for coming along today and listening in, as well as to the support from Opepe Farms, Tauhara Middle 15 Trust, and Tu Wharetoa Māori Trust for funding, for support, as well as our funding support from the Global Research Alliance on Agricultural Greenhouse Gases, MB's Strategic Science Investment Fund, and the National E-Science Infrastructure for Computational Support. And I will take questions next. Tēnā koe.
Many thanks, Manpreet. That was a great overview of some really exciting and very deeply technical work. I think people are still digesting what you had to say and thinking about what their questions are. So but I have one. So, what are the effects of the sulfur because you're doing that work in a geothermal area? When I drive through there, I'm more than anything, what I notice is the sulfur in the atmosphere. Is that important?
So geothermal sites vary a lot in the nature of the kind of thermal processes underneath. The particular site that we selected actually does not have a sulfurous component to it. It's a steam heated depression. And what that means, the soils are actually a lot similar and chemically and a lot more consistent with what we find everywhere else. And that's probably why this result is so interesting, is that we don't have these unwanted or unpredictable interactions with sulfur or any of the other additional compounds that are part of that natural geothermal process. So we were really lucky. We just have a steam heated depression rather than a more chemically complex geothermal activity.
Great. Good to understand that.
Yes.
Now, I've got one other question, just quickly before we ask some that are starting to come in. You talked about the enzymes making the changes to the microbes so that they perhaps can adapt to higher temperatures. How long does that take? Are we talking about something that happens over generations of microbes, and how long is a generation of microbes?
That is exactly the exciting question that we're interested in. In fact, that's part of what my background is about, is how long does it take for microbes to evolve and adapt? In the lab, you can grow a microbe and see it change within a few months. Microbial life cycles are very short. Their generation times are very fast. A single bacterial cell might take between 2 to 4 hours to replicate and produce its offspring. And that within that time, it can incorporate that information from environmental change that can affect its offspring. Now, that's a very simplified idea and comes from a laboratory understanding. What happens out in soil is a very complex environment where microbial growth is limited, environment is a lot more hostile and variable, which means that adaptation takes a lot longer. But still, it's possible that microbial adaptation is a lot faster than many other things that we care about. For example, plants, it's likely that microbes will continue adapting faster and adapt to the future environments better than some of our most valuable crops, for example, or forests, or the biological organisms, the earthworms, the insects, the birds. Microbes, in some ways, do have a better shot at adapting to climate change than many of the other species, let's say. I hope that answers your question.
That's been really great. And here's a kind of a follow-up question that's come from Suki. Even with evolution responses to temperature changes, there appears to be impacts to the biodiversity of microbes. Do we have an understanding of what that might mean for soil productivity?
Absolutely. I think that's sort of what we are seeing here. Not only the microbial identity, so not only which microbes are present, it's possible that what they will do will also change in the future. And that's sort of where we're leading to with this, that we need to be very careful around designing our future food crops, our future protecting land in a way that we can mitigate some of these massive changes. The microbes that we were seeing in this study specifically were all tending to be thermotolerant, really able to live in a hot environment, but they also had ability to live in an acidic environment, which again tells us something about what the future soil might be like. That not only it will be warmer, it will be more acidic, and that's again gives us an idea of what that function, how that function might change. One of the follow-up studies that we have planned looks exactly at that. Will these microbes have higher heat stress tolerance? Will these microbes have higher ability to live in an even more hostile soil environment? So yes, absolutely.
And I think also from that question, just another along the same lines, Kate's asked us, how do you get a change in the microbial biomass across the temperature gradient?
So across the temperature gradient, we didn't necessarily look directly at biomass, and we didn't quantify it, but through some magical statistics, we were able to estimate that overall biomass also decreases. And that links to that question around water holding capacity, that the more microbial biomass that's present is likely to create the organic material that's needed for having a higher water holding capacity. As that biomass declines towards the warmer and warmer parts of the gradient, we're losing the biomass, and potentially we're losing some of that water holding capacity potential. Our study site was interesting because we had all these trees with a nice thick layer, and as it gets warmer, all of that starts to thin out. All our sampling stopped before we hit bare rock, but basically that's where it's heading. So it slowly declines, and then you end up on bare rock, which is where the really, really hot steam heated depression is.
Great, thank you. Well, look, we've got some great questions. Graham's asked us, can we comment, or that's you, Manpreet, not me, on how will potential changes in the composition of soils impact the rates of biological or chemical changes within the soil? Even, very limited changes in average soil temperatures expected.
That is a massive question, Graham. I will try to comment on it. So temperature is one of those things that pretty much regulates all life as we know it, and a lot of that happens through mediating the cellular and enzyme kinetics. So every live organism is affected by temperature, and that's the most high level statement I can make without getting into trouble. What that means for biological and chemical reaction, it varies a lot. It varies from for a microbe all the way to plants. Certain plants will have ways of buffering against temperature up to a certain extent. Even small changes in temperature can often lead to these interactive effects on everything else, because soil and life just generally doesn't exist in isolation. It's like almost like a domino effect. If one part of the process is affected, it's likely to affect the next thing in line, and then the next thing in line, and then the next thing in line. So even small changes in temperature can affect biological activity, can affect chemical breakdown abilities, can affect enzyme kinetics. So all the way to life itself, all the way to reproduction and growth and everything in between. We think that in the very early stages, there will be this predicted improvement in growth as some plants and some microbes move into a slightly warmer environment. But as soon as that takes over, it's going to push past, at least for plants, push past the tolerance windows, and that's where the danger sits. In many places around the world, we're already seeing those dangerous and tolerable limits with temperature change.
Right, and I think we've got time for maybe just one more. And I think this relates to that feedback loop and so on that you're talking about there as well. What's the potential of the microbial genomes to evolve special mechanisms or DNA changes to deal with increased temperatures?
Oh, I think the potential is immense. Already from the work that we've done, we're seeing that microbes can adapt, microbes will adapt. Microbes have adapted in these geothermal hotspots to reflect their ability to tolerate high temperatures. The interesting question is, do those genomes still contain those other functions as well? We know these microbes can respire just about as well as the heated microbes can respire just about as well as microbes that are not heated right next door, but they look entirely different in their community composition. Their genomes look different, and we next need to drill down that genomic information to identify whether those functions also look completely different.
Thanks, Manpreet. Look, there are a couple of other questions and a few fairly complex ones as well. And I want to reassure everybody that's asked those questions that we will come back to you with some responses, but we'll do that in writing, just so that we've got a little bit of time to think about that. And that information alongside the presentation and recording from today will be available on our website in the next day or so. You'll also receive a link to watch the webinar again and share it with your colleagues. I'd like to say thanks from the whole team here at Manaki Whenua. I'd like to say a special thanks to Manpreet for a fabulous talk today, and we look forward to seeing you all again at our next webinars in May. Thanks. Bye.
Thanks, Lucy.